Now we discussed the controlled drug delivery system part 2, right? Previously we discussed the part 1. Here we discuss the definition and some terminology. Modify release drug delivery system. Drug release future based on the time course and location that are designed to accomplish therapeutic or convenience objective not offered by conventional release form. If the tablet or any device they are conventional, then they are not achieve the desired response, then we convert into the modified release drug delivery system. The modified release drug delivery system is divided into sustained release drug delivery system, controlled release drug delivery system, delay release, extended release, targeted release, and time release. Let's we discuss one by one. Sustained release drug delivery system. Here the technology utilized to slow release of drug from dosage form of delivery system over extended period of time. Only here we increase the release pattern, that is extended period of time. Okay. Now the control release drug delivery system. Here the technology utilized in which the rate of drug release is more precisely controlled compared to sustained release product, right? Control release system in which we are control the rate and time. It is also called as a predictable drug delivery system. Already we predict the dose and time, both are pre-predictable in case of control release drug delivery system. Next one is delay release drug delivery system. Those that use repetitive intermediate dosing of drug from one or more immediate release unit incorporated into a single dosage form, right? A conventional small dosage form, they are incorporated into the single dosage form, so they frequently release out from the device, but that all the small dose are in a single dosage form, right? So that is called as a delay release. Extended release, that allow the reduction in dosing frequency from that necessitated by conventional dosage form. Here only we extend the time duration. We are not consider the first order or zero order, only the time limited extended. Targeted release system, the drug release directed toward isolating or concentrating drug in body region, tissue, or site for absorption or drug action. So here that is particular targeting. The drug are targeted to the particular region and give the action, right? Next one terminology is time release. The release of the drug at a certain time period or duration of time. So Here we are consider the pulsatile drug delivery system. That is in osmotic drug delivery system. The drug is released out according to our need. So we are control the time period, that is called as a lag time. We are discussing in detail in osmotic drug delivery system. Next one is the factor which affect on the sustained drug delivery system or controlled drug delivery system. The first factor is physicochemical property. In that physicochemical property, we are consider the dose size, partition coefficient, aqueous solubility, and pK and drug stability. One by one we are discussing. The first one is dose size. For sustained greater amount of drug than corresponding conventional form. Already we are discussing in previous part. As compared to individual dosage unit, if we consider, then in case of sustained or controlled release drug delivery system, the amount of drug is more than individual dose in conventional drug delivery system, right? The drug require large conventional dose, that is unacceptable, right? If the drug require is amount is large, then it is the tablet or device is become a large and it is unable to swallow by the patient, so it is unacceptable, right? It is difficult to swallow by the patient. So the drug requirement is not cross the boundary of 1 gram. Okay, next one is same of drug require large release rate, but drug have very short half-life. So the frequent dosing is required. The drug release is large, but the half-life is very short. Okay, then limitation of drug. So for oral route, the patient acceptance, that means the drug requirement is not cross the 1 grams boundary, and then we are convert if into the sustained release or controlled release. For the intramuscular, vein or other route, the tolerance of drug at the site of injection. So that is the drawback, right? The next one is partition coefficient. It is defined as the ratio of fraction of drug in oil phase to that of the adjacent aqueous phase. So as you know, the partition coefficient is the ratio of concentration in oil to concentration in water. So here the CO is the concentration of drug in oil phase and CW is the concentration of drug in water phase. So the definition of partition coefficient K is CO divided by CW. The compound with high partition coefficient, that is predominantly lipid soluble and very low aqueous solubility. This can persist in body for longer time. That example is phenothiazine. So if the partition coefficient is high, means it is lipoidal in nature and very low aqueous solubility. So that is already it's remain longer period of time in our body. The compound with low partition coefficient then difficult in penetrating membrane because all our body part are lipoidal in nature, so that is poor viability. So already I always mention the three things must be required for the drug for better viability. One is the drug is always in a soluble form. Then second is drug have a sufficient partition coefficient or hydrophilic lipophilic balance, and the drug is always remain in anionic form. Then and then it is better candidate for so a better bioavailability. Next factor is aqueous solubility and pK. The influence absorptive behavior of drug. The aqueous solubility influences the dissolution rate as shown by the equation dc by dt is equal to kdA in bracket Cs minus Cb bracket complete. So here the K is the dissolution rate constant, A is the surface area, Cs is the concentration of drug in soluble form, and Cb is the concentration of drug in bulk fluid. The low aqueous solubility, the low dissolution rate, very low solubility less than 0.01 milligram per ml, then it is sustained, inherently sustained. For example, digoxin and griseofulvin, that drug have a very low aqueous solubility. Aqueous solubility of weak acid and base governed by pK of the compound and pH of the medium. For weak acid, as we know, the Hazelbach equation St is equal to H0 in bracket 1 plus Ka divided by H plus, or it is also represented by another equation S0 in bracket 1 plus 10 raised to pH minus pK. Here the S0 is the solubility of the unionized form, K is the acid dissociation constant, and H plus the hydrogen ion concentration. For weak base, St is equal to St SO in bracket 1 plus H plus divided by Ka, or it is another equation is S0 in bracket 1 plus 10 raised to Ka minus pH. Here the St is the total solubility of drug, S0 is the solubility of the free base form. As we compare these both equation for weak acid and weak base, here the Ka and H plus, they are replaced that place, right? So the solubility of weak acid and weak base is important or calculate during the manufacturing of CRDDS. Weakly acid drug in stomach in pH 1 to 2, that means in acidic media, it's remain anionic form, so the absorption is high. So acidic drug is remain anionic in acidic media, so absorption is high. On the other hand, the weakly basic drug in ionic form at the same site. If the weak base drug are present in acidic media, so it is ionized, so it is poor absorption. So in the formulation, often the vehicle is adjusted. So the vehicle is important during the manufacturing of sustained release drug delivery system or controlled release drug delivery system. The another factor is protein binding. The drug bind to more plasma protein than less elimination and act as a depot, which act as a prolonged release system. For example, the quaternary ammonium compound bind with mucin in the GIT. So here the only possible in reversible protein binding. If it is irreversible, then there is not we are consider a depot system. But if the reversible binding, then drug bind to plasma protein and it is reversibly, then depot system is occurs, and there gradually that reversible process is occurs, and drug is remain free in blood plasma, and the concentration of drug is remain constant. So it's already sustained release profile is obtained. Another factor, that is drug stability. The orally administered drug have two problems, that is acid base hydrolysis and enzymatic degradation. So the stability is considered during the manufacturing of controlled drug delivery system and sustained drug delivery system. For the drug unstable in stomach, so you are prepared the drug which is remain stable in intestine. So you are prepared the colon targeted drug delivery system. For drug which unstable in intestine, so prepare the protein drug delivery system. So here the drug which is unstable in intestine, to prepare the drug or dosage form which is remain stable in stomach in acidic media. So depending on that stability, you apply the approach. Unstable in small intestine, that is poor viability, that is here the drug which is unstable in intestine, the example is given, propanthelin and probenthine is the drug which remain unstable in small intestine. The next factor is the biological factor, that is absorption, distribution, metabolism, elimination, half-life, and side effect and safety consideration are considered during the manufacturing of sustained drug delivery system. The first factor is absorption. The rate of release should be much slower than rate of absorption. Then and then we expand the duration of time, and here we need expand the time duration in the treatment. Assume transit time in absorption area is 8 to 12 hours. The maximum half-life for absorption is 3 to 4 hours. So the apparent absorption rate constant is 80 to 95%. Lower absorption rate constant than poor candidate. The SR preparation absorbed by active transport or in a specific region of intestine. Disadvantage to the absorption, that is ferrous sulfate. So here the particular drug have a absorption window in particular area. So here the ferrous sulfate have a absorption property in particular region, particular area. So prepare according to that. You are apply the approach of manufacturing process according to absorption area. Some technique to maintain the drug in maximum absorption area is formulate the low density pellets or capsule or tablet, that is called as a floating drug delivery system, and the formulate with bioadhesive material, so they remain intact with the absorption site or where the maximum absorption is possible in the particular area. So you apply the bioadhesive material. Using the chemical penetration enhancer is the technique to enhance the bioavailability. The next factor is metabolism. The drug metabolized before absorption, then decrease the viability. So drug release at slower rate, the less total drug to enzymatic process and complete conversion to its metabolite. For example, allopurinol. So you know that in first part metabolism, most of the drug pass through the liver, and in the liver most of the enzyme are present which destroy the drug. In case of orally, right? Here the drug, when they bind with the enzyme, then here the enzyme try to metabolize the drug. If that drug dose is more than that receptor, that enzyme, then they are not bind to that area and they remain free in the blood and it's give the action. But here what happen, if we release the drug in a slower rate, so that drug which is low in quantity, so the enzyme are free to metabolize. So most of the drug are metabolized, so there is no free drug is available in blood, so no action you obtain. So the metabolic process is important in the manufacturing. Next factor is distribution, the volume of distribution, that is pharmacokinetic parameter. So very important, VD is very important, consider during the dose calculation. So in a dose calculation equation, the value of VD is required. The distribution of drug affect the elimination kinetic. When the drug is exactly distributed, according to that elimination rate are considered. So the two parameter, that is apparent volume of distribution and ratio of drug concentration in tissue to that of plasma, that is T by P ratio, drug concentration in tissue divided by the concentration of drug in plasma, that ratio is important. Apparent volume of distribution is predictor for drug dosing regimen. When you want to calculate the dose of drug, the value of VD is important. Our next factor is elimination and biological half-life. The rate of elimination described quantitatively by its biological half-life. The biological half-life can be calculated in case of first order release rate. T 1/2 is equal to 0.693 V by CL S, that is short half-life, that is excellent candidate. The ciprofloxacin that have a half-life 3.5 to 4.5 hours, then it is very good candidate for sustained drug delivery system. The drug having the very short half-life, that is less than 2 hours, not suitable. For example, furosemide and levodopa. The compound with long half-life, that is greater than 8 hours, so not suitable because it is already sustained. Here the digoxin, warfarin and phenantoin, which are the drug which have a long half-life, so it is not suitable for preparation of sustained release drug delivery system or controlled release drug delivery system. The next factor is side effect and safety consideration. So there is a therapeutic concentration range is listed. The side effect can be due to plasma concentration and overdose. So the therapeutic window is window and therapeutic index is very important during the manufacturing of sustained drug delivery system. So the sustained release system minimize the side effect. The measurement of the margin of the safety of drug, that is therapeutic index, TI, is equal to TD50 divided by ED50. TD, that is median toxic dose, and ED, the median effective dose. So that ratio of that median toxic dose divided by median effective dose is very important. It is also called as a therapeutic index. Is very important when you consider or manufacture a controlled release drug delivery system or sustained release drug delivery system. The small value of therapeutic index, that is potent drug, and high value of therapeutic index, that is safer drug. So if the drug is potent, then to note down that things, because if very fluctuation is occur in that therapeutic value, then it give the very toxic effect in case of potent drug. So when you prepare the sustained drug delivery system in with the drug of potent drug, that is very important for the consideration. Role of next is the role of disease state. The disease is equally important as drug property in consideration of SRDF. The nature of the disease is the stimulus for the development of SRDF. Aspirin in rheumatoid arthritis is considered. Only we are consider the patient is healthy in case of calculation of dose in sustained drug delivery system. So that is the drawback. The drug release pattern is altered in different disease, that is in kidney failure, hepatic failure, or in particular disease. So the better way is select the particular patient for the particular drug development is the best way, right? For example, when we prepare any sustained drug delivery system with the help of aspirin medicine, at that time use the patient who suffer from the rheumatoid arthritis, right?